Okay, my friends, we're going to talk about Alzheimer's, autism, all the neurological disorders, primarily that are up in your brain area. And they're relating it to tau proteins. And they don't break down. They collect and they clog things up and cause issues. Now, I think there may be a solution to that, which is not going to be a hard solution, but it needs to be investigated, and I'll show you exactly how it can be done. And it's not a big thing to do, but we have to look at the enzyme portion of this process, because I'll explain it to you as we go through the chemistry. And it's not a big deal. Don't get freaked out. Everybody, chemistry, I'm scared of that. No, don't be scared of it. I'm going to show you how bacteria create these enzymes. And they do the job of everything in your body. Literally everything in your body is done by an enzyme. 100%. Zero happens that doesn't involve an enzyme. Can't happen. An enzyme creates the chemistry so fast to make the process work. And they also create products, proteins, and all kinds of things to make your body run. And those are done by bacteria. Bacteria are the most important thing in your whole body. Let's get started. All right, we're going to look at this tau protein, because that's what they're talking about, is it doesn't break down the way it should. And it consists of 4 repeat domains and a C-terminal, so forth. And its repeat domain, it stabilizes these microtubules, preventing them from falling apart. This is all that means, is falling apart, depolymerization. Now, so it normally stabilizes the microtubules so that they work correctly. Now, tau protein also interacts with other proteins involved in microtubule dynamics and neuronal signaling. Well, if you can't get rid of the tau protein when it's supposed to be, taken to the trash, then there's a buildup. And they say that's the problem. All right, remember this now. Tau is a protein. And it's good. It's supposed to be there to stabilize the microtubules. But when you don't get rid of it, it just collects. Now, they provide the protein coding genes, which we only have 25,000, whatever it was, provide the instructions for building proteins, right? Building proteins. Those are the building blocks of life. They're which are essential for nearly every cellular function, contribute to an organism's traits. The process known as gene expression. This is important because genes can be there, but if they're not expressed, they don't work. And that's something called methylation. We'll talk about that later. So the gene is there, just waiting to be turned on. If it doesn't turn on, it just sits there. It's just like a switch. You need something, turn the switch on. But you have to demethylate the sequence to say, okay, now it's your turn to do something, and then it turns on. So that is the expression. And it involves working with the messenger RNA molecules and translating that into a specific sequence of amino acids, which are the little building blocks. They form a protein. They go out and do a job, and they do it fast. And this is all about building the proteins, protein functions, transcription, and coding. You don't have to know a lot about this. But what we're going to be talking about is ribosomes a lot. All right, well, let's just read this through. Simple. Encoding instructions. Gene is a segment of the DNA, just a little chunk. It contains the code for a protein. The order of the nucleotides within the gene determines the specific order of amino acids in the resulting protein. So the nucleotides are the little bits and pieces inside that segment, just like a program. Transcription. The first step is transcription, where the DNA sequence of the gene is copied into a little chunk, or half of it basically, which is the messenger RNA molecule. Translation. The messenger RNA molecule then moves to a ribosome. All of these things that happen are done because of an enzyme facilitating that reaction. These aren't just going to happen automatically by themselves. There's an enzyme involved in every one of these things. Now, this mRNA molecule moves to a ribosome. I'm going to show you this in anatomical in a second. It's translated in that ribosome. The ribosome reads the mRNA sequence in 3 nucleotide units, codons, each codon specifically a particular amino acid. Now, building the protein, the ribosome links, again, ribosome. It links the amino acids together in the order specific to do the program. It's the program, forming a polypeptide chain that folds into a functional protein. So it gets in a little tiny thing inside the ribosome. Once it hits its target, it goes into the 3D. The final protein function, the final 3D shape and function of the protein are determined by its amino acid sequence dictated by the original gene. So the original gene had programmed the sequence in order, and when it popped open, it's going to be exactly the same every time, because they link all of these little chains, these polypeptide chains together in an exact certain way, and then they put them all together in a tiny little ball called a ribosome. Then when the ribosome triggers, it goes into whatever the very elegant structure is. Again, I will show you this momentarily. And it's dictated by the original gene. The proteins are vital for tasks like catalyzing reactions, which every single thing in your body is a catalyzed reaction. It doesn't happen quickly enough to support life. All right? It's providing structural support, transporting molecules, breaking things down, digesting. Everything is done by one of these enzymes. And an enzyme is a product of this sequence coming out of a ribosome. Now I'm going to go into some chemistry. It's not going to be a big deal. If you really want to understand this, you got to dig a little deeper than just surface. You would die almost instantly in a matter of seconds without enzymes. How fast would you die without enzymes? Without enzymes, the biomechanical reactions necessary for life, such as everything, respiration, metabolism, everything, be too slow to sustain functions. You fall over dead. Listen to this. Life as we know it requires enzymes to speed up reactions, which normally would take billions of years to complete that reaction on their own if you didn't have the catalyst. Billions of years. Done like that. Less than a second. And it goes on from one to the next to the next. You don't even use up that enzyme. It stays working forever. And this is 2008, he knew about these enzymes. All right, when you're born with DNA. After that, and there's only a certain very small percentage of your DNA is to code. In other words, it's like they used to call it junk DNA, and I think it's like 98% or even more than that. Is they don't know what it does. It's just they think it's junk. It's not junk. It's there to facilitate your understanding of life, your learning, your emotions, your senses, your feelings, everything are on there, and your memories and all that. Because it's just a stunning amount of memory. And they don't realize how much memory there is in the DNA. But anyway, you do have to understand a little of this stuff. And but once you understand dipole electron flood theory, the rest becomes fairly simple. And right now, enzymes are the things that are doing the half-lives. And as I showed you, they can do things, 1 billion years worth of chemistry in less than a second. And the only way you get those enzymes are from bacteria. That's where they come from in your body. And once you damage your bacteria, it has to repopulate. And if it can't repopulate, you have these long events of chronic diseases. Here it is. Bacteria act as factories. The bacteria is the factories. It's not the organs. They say, oh, the pancreas does this, the gallbladder does this. No, it's the bacteria that live in those areas that create the enzymes. Listen to what it says. Bacteria act as factories. The bacteria squirt out those ribosomes, which are the enzymes, because they use genetic instructions. So they have a program. It's the genetic instructions of a program to produce and secrete a vast range of enzymes to break down food and other complex materials. They don't just break things down. They do all kinds of stuff in your body. They create all the chemistry that you need to have to do everything that you do. And without enzymes, you're done. All right, so this is where their focus is on this tau protein. And I can understand that because there's more than should be there, so you should get rid of it. Now, abnormal aggregation, that means just balling all together, and phosphorylation of the tau protein, and we're going to look at that phosphorus, of the tau protein are key features several neurodegenerative diseases, including Alzheimer's disease, frontotemporal dementia, Pick's disease, and I believe there's a lot of them. But let's look into what is phosphorylation? What does that mean? All right, to cut to the chase quick, we're talking about genes. All right, well, here's a sex chromosomes, 6 billion. Now, in the human genome contains about only 20,000 to 25,000 protein coding genes within the approximately 3 billion base pairs of a haploid set. All right, so we're only using 25,000 protein coding genes. Well, what's a protein coding gene? What does that mean? All right, this gets a little confusing, but the codons and amino acids. The codons are not amino acids. The codons are these 3 nucleotide unit things that come down to program the mRNA, which is a sequence that creates a specific amino acid to link together. And here it is, like I say, it's a little confusing. You don't have to know that. Who cares about that? It doesn't make any difference. You see this cloud here? That's transfer RNA, transferase, they call it. It's an enzyme. And each one of these coming in is coming in from the bacteria. The bacteria has a special DNA code that's extra from the DNA code of its own self. It has an extra DNA code, which is here, that creates the ribosome. And it does that by shooting out each one of these little chains in a certain order. And these things here are the transferase, and they transfer these into an mRNA string, messenger RNA. And then it goes and breaks into a DNA. Now, this is how you become immune to things. Your body creates this sequence that goes up and cuts in using uracil, I believe is the way it snips in, and then it shoots it into your DNA, and it must transport it throughout your DNA, and you become immune to certain things. But I also believe this is the way that you can become seriously ill as well, unless you have the correct bacteria in here to make enzymes to do the job to fix that bad sequence. Okay, you remember I showed you these little balls? All right, those are ribosomes. All right, and these are right up here. These are the ribosome balls. They come out of bacteria, and they just squirt them out, floating around, squirting them out all over the place. And I think they must just squirt them out like a hose, because we use a lot of these ribosomes. And guess how small these puppies are? You never imagine how small they are. Inside of that ribosome is a chemistry set which is just so elegant, it's amazing. Inside each one of those ribosomes is something like this. It doesn't pop open like that until it gets a trigger. All right, so this, in each cell, one single cell in your body, one single cell like this, ribosomes are in there just everywhere, and primarily they collect on this rough endoplasmic reticulum, basically to protect the nucleus, because they're very chemically active. They're extreme. Enzymes are unbelievably elegant molecules. Inside that one single cell, with all the other things it's got going on, the mitochondria, to this, to that, to that, in addition to that, we can have up to millions of ribosomes. It's still in that one cell. Can you imagine that? And when they pop open to something like this. To do, every one of those is a specific magnetic moment. And when it's called click chemistry. It's almost like electrocution. Whenever it hits something that it is programmed to destroy or break or do whatever it has to do to it, done. And that literally would take, in some cases, millions of years to do if it wasn't for the catalyst. It can never happen fast enough without one of these enzymes. So basically everything that's going on in your body right now requires enzyme. They're going. And here they are. This is how it works. There's the enzyme. It comes in and there's a little foil around it or something, and the sheath comes off and it does this. Now, are enzymes proteins? Well, yes and no. They come from the same source, from a bacterial ribosome. That I think most people would agree on. However, proteins build things. Those are building blocks. These are destroyers. These are jackhammers and sledgehammers, and they come in and they break things up. An enzyme is caustic and it can break things up, and it does all that stuff. These build things. So these break them, and they go over to here, and these guys build things up with them. You have to be able to break down a carrot. You can't eat a carrot and it just turns into a muscle. It just doesn't happen. All right, so that's what's going on. And phosphorylation is the key to this whole thing. It's all about membranes. All right, when we're talking about this phosphorylation, reversibility process can be undone by enzymes called phosphatases, allowing the cell to turn a signal on or off as needed. Well, we apparently have either too many of them just collecting and not being broken down, or we have too much production. It's one or the other. But enzymes are going to be the solution to this, other than toxins. Toxins, you can make them break down probably with some toxic substance, yes. But enzymes will do that job in a non-toxic way. All right, the whole reason we're talking about this is because they claim that this hyperphosphorylation of the tau protein leads to dysfunction and aggregation, collecting, as I talked about before, and tangles. And that's the hallmark of this disease. Now, this is reversible with enzymes. I think they say that it's reversible with enzymes. All right, here's what it says right here. Phosphorylation process is reversed by phosphatase enzymes. Those are these really sophisticated enzymes. Where do they come from? They come from bacteria. They claim that it's controlled by the phosphorylation, dephosphorylation of ribosomal factors. Ribosomal, they come from bacteria. Control protein synthesis in response to cellular signals. Well, I don't think it's the cell that's giving out the signal. I think it's the bacteria that's giving out the signal, but you have to have the bacteria. And the bacteria are in there to sense issues and then to deal with them. They do both of those things. And this has been observed. All right, this is what they've been working on right now, is this tau protein. And it becomes hyperphosphorylated. This is in Alzheimer's disease. Enzymes are supposed to control that protein. All right, and do enzymes control activity of these tau? Yes, enzymes control tau activity. It's a reversible process. You need this tau, but you don't need it in excessive amounts. They're talking about hyperphosphorylation. Anyway, I'm going to go on to explain this, but I think we should be looking for some enzymes. And I would like to see a study done on fecal matter transplants to see if the enzymes, which are the product of the bacteria in the fecal matter, that's the bacteria, that's what you need to make these enzymes. I would like to see this study done on that to see, does that help? Because as I will show you, it's all about controlling these enzymes. I mean, having enzymes to control these activities. They're not being controlled well now. And I think as you age, the bacteria ages with you, possibly. And, you know, a good new set of bacteria in your gut will hopefully help even in the older people. And it appears it does. There's been some studies, or not studies, basically, just observations. And that's what I do, is observational studies. And they're studies, but they're observational. I've been doing it with over 5 years with people in Belfast, Ireland, Northern Ireland, on autism. And it's been very, very successful. Very successful. At basically reversing some of the symptoms, and most of them, going from completely 100% in the worst side of the spectrum back to being on the very minimum side of the spectrum, if at all. And that's by changing the environment of the gut. It has to be a nice, clean digestion in the gut, and getting no problems with the gut. You got to have your gut under control. You're good to go. Hippocrates said that 2,000 years ago. All right, this should be worked on immediately. This is nothing more than the bacteria that's in people's digestive systems. Key observations from studies, cognitive and clinical improvement. One case study reported significant improvement in a patient's mental acuity, memory, and mood after the fecal matter transplant for C. difficile, which is a gut infection. The patient's mini mental state examination score improved from a low point to near normal. Other studies, including preclinical ones, have shown that FMT can improve spatial recognition memory and reduce amyloid beta plaques in the brain. You need the enzymes. You only get the enzymes from the bacteria. And this is from the changes in the gut microbiome. Impact on disease. FMT appears to affect pathways linked to Alzheimer's, such as reduction in inflammation, accumulation of these plaques. It also influences the levels of short-chain fatty acids like prophylactic acid and butyric acid, which are important for brain health functions. Limited clinical data. Currently, there are very few clinical studies. Nobody's doing it because it's not something they can make a ton of money on, is my opinion. More large-scale clinical trials urgently needed. While the connection is promising, the exact biological mechanism improved symptoms are still being investigated. It's the enzymes from the bacteria, which is in the microbiome. It absorbs it, and it can break things down. Without the enzymes, you're done. It's all about the enzymes. And it is promising because it is the enzymes, they have to be there continuously. I mean 24/7. You use up enzymes apparently very quickly. because you can get very sick in a very short amount of time if you have your enzymes damaged, and that's easily understood by seeing acute radiation sickness, where they get hit with a radiation pulse and the bacteria in your body die. Basically, the bacteria is the stuff that gets killed. And then about 2 hours later, you start throwing up and having diarrhea and everything else. Your digestive system, done. Bacteria is not as strong as the rest of your body cells. Your body cells are pretty protective against radiation compared to a bacteria. So I'm telling you right now, we need to do these microbiome transplants. It's as simple as that. And so let's start with that. And if it doesn't work, well, it didn't work. But why is it always this, we need more, we need more, we need more? This goes back years and years and years. I've been talking about this for 10 years, at least. All right, here's the conclusions. Their synthesis of this demonstrates that the fecal matter transplant exerts therapeutic effects by modulating this AB metabolism and tau phosphorylation. It modulates it. It reacts with it. It restores the neuroendocrine equilibrium. That's what you need, is the equilibrium, attenuating neuroinflammation, enhancing intestinal and blood-brain barrier integrity. It takes care of basically everything, because you have a new bacteria creating new enzymes. While preclinical studies demonstrate this remarkable potential in alleviating Alzheimer's disease pathology, clinical translation requires methodically rigorous randomized controlled trials to establish their efficacies and safety profiles, and they are just not doing them. You see this? It's hard to believe this is not being taken care of. Rapid improvement in Alzheimer's disease symptom following fecal transplant. This is 2020. That's 5 years ago. I just showed you one that just came out in August 2025. Same thing, but we need more research, more research. All right, this goes back to 2015, 2020. I've found them going back to 2000. They know about the benefits of getting more bacteria in your gut and how devastating antibacteria things that kill the bacteria in your gut are. The conclusion of future directions. This is, I think, from 2023. Based on the above evidence, we conclude FMT beneficial for Alzheimer's disease, which has a serious impact on the health and quality of life on a patient. It's urgent to find low-cost and few side-effect intervention measures, which this is. This is hard to believe that it's not being taken seriously. This goes back a long time. They've known about this, and for some reason, it needs more research. We need more time. We need more studies. First, you have to understand about phosphorus. You see this? This is the cell membrane. Around every cell in your body has a membrane around it. And every organ, they all have membranes everywhere in your body. And if you come up to here, that's that little tiny layer. You see this phospholipid bilayer. Phosphorus on the top and on the bottom. Phospholipid. Lipid means fats. Phosphorus and fats make up this layer that separates that cell inside from everybody else. And then you have a whole bunch of cells stacked together around an organ or so forth, and that's another membrane. So I'll show you that in a second. Well, I'll show you right now. It's this right here. These are cell membranes. Different types of cell junctions, and these are the junctions between the membranes, and those would coat organs and so forth. And these are, tight junctions. You don't want any leakage through there, because then you get invaded, and then you get what they used to call cancer. Stage 1, 2, 3, 4, and finally it breaks through that layer, and then it just starts living off of you. It's trying to get down here to get something to eat. Basically, that's it. Think of it that way. It's a little living thing, And it's trying to get down to get something to eat. And once it gets through here, if you can't stop it, then you're in trouble. And that's what cancer is. And all chronic diseases have the same similar patterns. And all different areas of your body have different types of membranes. And it depends on where you are. If you're in the digestive system, or, you know, all through your body you have different, especially in the digestive system, and where you eat, and whatever passes through you has to be, you got to deal with it, because you're going to have some nasty stuff. You're going to get some nasty stuff coming through you. And this is what deals with it, is your membranes. And a child, when the digestive system of a newborn there's really nothing there when they're newborn. They need to program. And that programming is the bacteria that they're associated to, and the environmental toxins, everything. They have to live in the environment that they're born into, and that's pretty much, you know, hopefully it works out. Sometimes it doesn't. Sometimes they don't have the right genes. I agree with that. You can have all kind of problem with genes. And I think there can be more involved problems than they're thinking about, because they think every molecule is one big ball of just one molecule. No, it's a zillion little tiny molecules or dipoles together make up that molecule. So there's much more complexity to every molecule, like amino acids and so forth. They see them as sort of, they're complex, but they're certainly not as complex as they really are. That's just, chemistry has to change, because these things, like, in other words, let's just say hydrogen, it's not one. They say it's one, like this. It's not one. It's about 1,840 dipoles, and that can be split into 1,840 different pieces. All right, so we saw about biology. We got some issues there. We got some issues with physics. We got issues right across the board in science. So we got to start talking about things. But this is what the dipole electron flood theory is, and this is dark matter. And I've shown all the experiments over and over and over. So I'm going to leave it at that for today. But stick around, stay tuned in if you want to learn reality. And I'm telling you, this you're learning what has been unknown. This is unknown. It's not unknown to anybody. And the ones that are laughing at it, well, good for them. But you're learning if you're learning and paying attention. And then we can all discuss this together. And like I said the other day, this is, right now, dipole electron flood theory is 100% accepted in the scientific community. I have bounced this off of virtually everybody, and nobody contests it. That means it's 100% accepted. All right, I love you all.